Okay, hi everyone. This is our first video of our MCAT series where I'm going to explain the material covered by the Kaplan MCAT books since I'm using them to prepare for my MCAT. Today, we'll be going over Biochemistry Chapter 1, which is amino acids, peptides, and proteins. Section 1 is talking about amino acids found in proteins. So what amino acids are, they are just a molecule that have an amino group and a carboxyl group. What makes them unique and determines their chemical properties is actually the variant group, which is the R group. All the amino acids in eukaryotes are L-amino acids. So what that means is that the amino group is on the left side in the Fisher projection. You can see the D-amino acid is the mirror image of the L-amino acid. L-amino acids translate to S-absolute configuration. There is an exception. Cysteine, however, is an L-amino acid but has an R-absolute configuration. So we can just write it cysteine. And that's because the CH2SH group has a priority over the carboxylic group. All amino acids are chiral, except glycine. So we have three special cases of amino acids, and we already discussed two of them. So now we're going to move on to the third one. The third one is proline. Proline is unique because the amino group is on the R group. So let me just explain that. So as you can see, the alpha carbon, the carbon in the middle, is called the alpha carbon. And what that means is that the amino group, the variant group, and the carboxyl group are all connected to that carbon. You can see it in every amino acid except proline. So right here, that would be the alpha carbon, alpha carbon, alpha carbon. You can see that the amino group, the carboxyl group are connected to it. But on proline, the nitrogen of the amino group is actually on the R group. That makes it unique and very useful, which we're going to talk about later. For the MCAT, you do have to memorize all of the structures of the amino acids, plus the 3 and 1 letter abbreviations. We have 5 amino acids that are charged. They have charged side chains. So right here is arginine, histine, and lysine, which they have a positive charged side chain, and that makes them basic. And we have aspartic acid and glutamic acid, which have a negative side chain, and that makes them acidic. But the MCAT is not going to use those names. According to the Kaplan book, they're going to use aspartate and glutamate, which are the anion names for those. As you can see, the R group of histine has a ring, and the ring is actually called imidazole. 7.4 pH is the physiological pH of our bodies. We already discussed the special cases, and these are the 20 amino acids that you have to memorize for the MCAT. There are actually 21st and the 22nd amino acid, but according to the Kaplan books, they are not going to be on the MCAT. Which the 21st and the 22nd amino acids are actually cellulose, cysteine, and pyrrolysine. These are just stop codons. If you take any genetics class, you should be able to learn more about these. There is a website that has a chart with all the amino acids in it, and it states the hydropathy of them and the size of them. So it's a great source to just memorize all the amino acids that you need to memorize. I will link the website down in the description. Amino acids with long alkyl side chains are strongly hydrophobic. So that means they don't like water. They cannot mix with water. And those amino acids are actually alanine, valine, isoleucine, leucine, and phenylalanine. And since they are hydrophobic, they are in the interior part of the protein. Amino acids with charged side chains plus asparagine and glutamine are strongly hydrophilic. So they would be on the surface of the protein interacting with water. So at the end of every section, I'm going to put questions related to that section, so you can review it later on just to active recall. So here are the questions for this section. Section 2 is about acid-base chemistry of amino acids. Amino acids are amphoteric. So what that means is that they can either accept or donate a proton depending on the pH of the environment. So if the pH is higher than the pKa, that's the pH of the environment. If it's higher than the pKa, they will lose protons. They will deprotonate. If the pH is lower than the pKa, means the environment is very acidic, full of hydrogens, they will gain protons or gain hydrogen. The pKa of a carboxylic group is around 2 to 3, and the pH of the amino group is around 9 to 10. So what that means is that we got an example here. At a very low pH, so let's say at a pH of 1, we can see that the amino group and the carboxylic group are both protonated. And if you move to an upper pH, for example, pH of, let's say, 5, we can see that the carboxylic group loses the hydrogen. So that's because the pH of the carboxyl group is around 2 and 3, and now the pH is higher. So just as we said before, if the pH is higher than the pKa, it will deprotonate. And then if you move even to a higher pH, more basic environment, for example, 13, pH of 13, the amino group will also deprotonate. So you can see here it lost the hydrogen that it had. So now it's NH2, and it was NH3 at first. Okay, now we're going to discuss the titration of amino acids. So we have glycine right here, and we are increasing the pH of the environment. We can see that we have some horizontally flat lines, and we have one vertical flat line on the graph. So we're going to discuss each of them. So we can see that the first flat line, horizontally flat line, is around the pH of 2.34. And what that means is that on that line, the carboxylic group actually gets deprotonated. So we discussed that the carboxyl group, the pKa of it, is around 2 or 3. And if the pH goes above that, the carboxyl group will lose a hydrogen. Okay, next we have the isoelectric point. It's the pH at which the molecule is electrically neutral. So at the pH of 5.97, the molecule is neutral. And if we keep increasing the pH, the amino group will also lose its hydrogen. So we can see here, after the pK2, if we increase the pH, the amino group loses its hydrogen, and also the carboxyl group lost the hydrogen at the pKa of 2.34. So we do have an equation to calculate the isoelectric point, which is right here. So if you add 2.34 with 9.6 and divide them by 2, you will get 5.97, because that's the point which the molecule is electrically neutral. Now we're going to talk about the titration of amino acids with charged side chains. So that matters because we're going to have a side chain which is involved in the net charge. So as you can see in the glutamate right here, we are having 1 pKa at 2.91, and pKa-R, that's the pKa of the side chain, which is 4.25. And then we have the pKa2, which is the pKa of the amino group, at 9.67. So what that means is that if the pH goes above 9.67, the amino group will lose the hydrogen that it has. And also that same goes for the R group and the carboxyl group. And we have the equation for both of them right here. This is for the acidic amino acid, and this is for the basic amino acids. And last point on this section is that the amino acids with acidic side chain will have a pi value below 6, and amino acids with basic side chain have pi values above 6. So this will help you a lot. Just to, if there's a question relating to this, you can quickly eliminate the wrong answer choices. So just should remember that. So now I'm just going to put up the questions for this section.